biotechnology application in different science. Please attention. Okay. One of the most important enzyme after recession endonucleases are ligase. پس یکی دیگه از مهمترین آنزیم هایی که بعد از آنزیم های محدود کننده مورد استفاده قرار میگیره آنزیم های لایگیز، آنزیم لایگیز هست. DNA ligase, the study of DNA replication and repair processes lead to the discovery of the DNA joining enzyme called DNA ligase. DNA ligase catalyze formation of phosphodiester bond between 5' phosphate of nucleotide on one fragment of DNA and 3' hydroxyl of another. This joining of linear DNA fragment together with covalent bond is called ligation. As you see, excuse me, can you hear my voice? بچه دوستان صدای من دارید؟ Excuse me for a problem in my microphone. خدمت شما گفتم یکی دیگه از مهمترین آنزیم هایی که در واقع در علم بیوتکنولوژی حائز اهمیت هست آنزیم لایگیز که در واقع بعد از آنزیم شدن اندونوکلاس ها یا آنزیم های محدودگرم مورد استفاده قرار میگیرند. As that, the most important enzyme after recession endonucleases is DNA ligase, and DNA ligase is used for very important in biotechnology because this enzyme joining two strands of DNA together. Okay, this enzyme help us to joining DNAs. Two pairs of DNA together with phosphodiester bonds. The study of DNA replication and repair process lead to the discovery of a DNA enzyme called DNA ligase. Okay, therefore DNA ligase is very important in biotechnology. Ligase catalyze formation of phosphodiester bond between 5' phosphate of nucleotide on one fragment of DNA and the 3' hydroxyl of another. This joining of linear DNA fragments together with bond is called ligation. It's called ligation. Unlike the type 2 restriction endonucleases, DNA ligase require ATP as cofactor. What is the difference between nucleases and ligase enzymes? The difference is the most important between restriction endonucleases and ligase is that type 2 restriction endonucleases doesn't need to ATP as a cofactor, but DNA ligase require ATP as a cofactor. Okay? Please see again.
Restriction endonucleases are enzymes that cleave DNA at specific nucleotide sequences. The sequence recognized is often 4 to 6 nucleotides long. For example, the restriction endonuclease EcoR1 recognizes the sequence GAATTC. The nucleotides at one end of the recognition sequence are often complementary to those at the other end. The two strands of the DNA duplex have the same nucleotide sequence running in opposite directions for the length of the recognition sequence. Because the same sequence occurs in both strands of the duplex, the restriction endonuclease can bind to and cleave both strands of the DNA molecule.
Okay, as you see that the restriction endonucleases cut DNA strands with two shame, with two figure as blunt end and sticky end.
Because the leaf is typically not positioned in the center of the recognition sequence, and the DNA strands are antiparallel, the cut sites are offset from each other. After cleavage, each DNA fragment has a single-stranded end a few nucleotides long. The stranded ends of the two fragments are complementary to each other. These single-stranded ends can pair with each other, sticky ends. Once their ends have paired, the two fragments can be joined together with the enzyme DNA ligase, which reforms.
As you see, DNA ligase, which reforms the phosphodiester bonds of DNA. DNA ligase reform the phosphodiester bond of the DNA.
Forms the phosphodiester bonds of DNA. What makes restriction endonucleases so valuable is that any two fragments of DNA produced by the same restriction endonuclease can be joined together. Restriction endonucleases are fundamental tools in genetic engineering.
I hope you found everything about. I think you understand everything. Understand everything about DNA ligase. Do you have a question about DNA ligase? Because it can join two pieces of DNA, DNA ligase became a key enzyme in genetic engineering. If restriction digested fragments of DNA are placed together, if restriction digested fragments of DNA are placed together under appropriate conditions, the DNA fragments from two sources can annihilate to form recombinant molecule by hydrogen binding between the complementary base pairs of the sticky ends. Complementary base pairs of the sticky ends. However, the two strands are not covalently bonded with by phosphodiester bond. Okay, therefore, the DNA ligase help to joining two strands of DNA with phosphodiester bonds. DNA ligase is required to seal the gap. Covalently bonding the two strands and regenerating a circular molecule. The DNA ligase most widely used in the lab is derived from the bacteriophage T4. This is very important for final exam. My friends, the DNA ligase most widely used in the lab is derived from the bacteriophage T4. And this figure is very important for final exam. This figure show you the mode of action of the DNA ligase, and it shows that DNA ligase how it works, how it can work. The phosphodiester bond between 3' hydroxyl from one strand to phosphodiester to phosphoric acid 5' from other DNA strand, and joining these two DNA strands together. Please write this information in your notebook, and you have 2 minutes for writing this information in your notebook. And please note that this information are very important for final exam. 2 دقیقه بچه شما وقت دارید این اطلاعات رو توی دفترتون بنویسید و حتما کنارش هم ستاره بزنید و بنویسید خیلی مهم برای امتحان پایان ترم. Okay. T4DNA ligase will also ligase fragments with blunt ends. Please attention. T4DNA ligase will also ligase fragments with blunt ends. But the reaction is less efficient, and high concentration of enzyme are usually required in vitro. To increase the efficiency of the reaction, researchers often use the enzyme terminal deoxynucleotidyl transferase to modify the blunt ends. Again, to increase the efficiency of the reaction, researchers often use the enzyme terminal deoxynucleotidyl transferase to modify the blunt ends to ends. For example, if a single strand polyadenosine tail is added to DNA fragment from one source, and a single strand poly-DT tail or polydeoxythymine tail is added to DNA from another source. The complementary tails can hydrogen bond. Therefore, recombinant DNA molecules can then be created by ligation. As you see, poly-T from the one strand end to one added to one of the DNA strands, and poly-A to added to another DNA strand with enzyme as deoxynucleotide transferase. Therefore, two blunt ends change to a sticky end, and these sticky ends can join together with a high efficiency. Okay, therefore this slide is very important. Please write this information. Writing this information in your notebook, and please note that this enzyme, what is the application of the deoxynucleotide transferase for efficiency of joining two DNA strands in biotechnology. Please writing this information in your notebook, and this drawing this picture in your notebook. These are very important. Okay, now I want to teach you gene cloning is very important section of biotechnology.
A little scared? You might be wondering, is it a tomato or a potato plant? It's actually both. Yes, you heard me right. In this single plant, the tomatoes grow above the ground while the potatoes grow below the ground level. And how is this made possible? It can be done by a process called grafting. In this, the stems of both the tomato and the potato plants are chosen and then joined together by tying them. This is an example of plant engineering. On the similar lines, when we cut and join genetic material, the process is known as genetic engineering. It's one of the principles of biotechnology. Now let's explore more about these principles in detail. Most of the principles of biotechnology are based on two engineering branches, that is, genetic engineering and chemical engineering. Genetic engineering is the branch which deals with the modification of the genetic material of an organism. And which genetic materials are we referring to? We know that it's the DNA which is the genetic material for most of the organisms. And what about the remaining ones? Well, in them it's the RNA which acts as the genetic material. Just like in the case of some viruses. So genetic engineering deals with the manipulation of genetic material. Moving ahead with the next principle, which is chemical engineering. The name itself suggests that it revolves around the use of chemicals for the growth and the maintenance of cell cultures. For this, desired eukaryotic or microbial cell cultures are stored in a contamination-free zone. Further, these cell cultures are used for the production of vaccines, antibodies, enzymes, and various other useful products. We will learn about chemical engineering in the upcoming videos. For now, let's just discuss in detail about genetic engineering. Did you know that the first ever genetic modification was done in a bacteria? That's right. And later on, these modifications were applied to animal as well as plant cells. The genetic modification can be done either by adding something new to the existing genetic material, or it can also be done by deleting something from the existing genetic material. The DNA obtained after modification is known as recombinant DNA. Genetic engineering can be done in many ways. Recombinant DNA is one way to pursue it. When genetic engineering is applied to modify organisms, they are known as genetically modified organisms, abbreviated as GMOs. And what organisms can be modified with this technique? Almost all living organisms can be modified with the help of genetic engineering. What kind of applications does this serve? Modifications in organisms can give rise to beneficial products. Consider plants, for example. With the help of genetic engineering, we can produce robust crops, pest-resistant varieties, and also high-yielding varieties of many crops. This was, in short, about one way in which genetic engineering can be carried out. The other way to perform genetic engineering is by making copies of the genetic material. This is also known as gene cloning. In simple words, gene cloning can be defined as the process of making an exact duplicate of the genetic material.
But why do we need a duplicate of the genetic material? Let's consider an example to understand this concept. Imagine there's a new product launched by a company in the market. This has some really cool and amazing features which will be of great use to all. Now, is it fine if the company manufactures just one piece? Of course not. There will be so many people who will want to buy it. For this, there will be multiple pieces of the same product manufactured on a large scale to suffice the demands. This just means that making exact copies of the original product is useful in several ways, isn't it? This same logic applies for genes as well. Genes which code for certain extremely important proteins or even the newly modified genes can be copied several times. This process of making exact copies of genes is called gene cloning. And just as in this case, here as well, clones or copies of genes serve many important applications. This process of making copies of the genetic material falls under the umbrella of genetic engineering. So in this video, we learned about the basics of genetic engineering. To learn more, watch our upcoming videos. Till then, just subscribe to our channel to get notified.
Okay, my friends, in this video, I show you everything about gene cloning. Do you have a question about gene cloning? بچه در مورد جین کلونینگ دارید یا نه؟ همچون که می‌دونید جین کلونینگ اصلا کلونینگ به معنی در واقع چیزی در اندازه‌های خیلی زیاد. اوکی، تکثیری در اندازه‌های خیلی زیاد. وقتی که شما یک صفحه کاغذ رو تکثیر می‌کنید، در واقع دارید اون صفحه کاغذ رو کلونش می‌کنید. پس جین کلونینگ یعنی تکثیر چیزی در اندازه‌های خیلی زیاد. این فیلم رو هم ببینید لطفاً.
Today, we'll talk about the process of recreating recombinant DNA. This lesson was made by Tyler Hogue and Mariah Thrush as part of Ohio University's NSF-funded Boat of Knowledge in the Science Classroom. What is recombinant DNA? Technical definition is DNA molecules formed by laboratory methods of genetic recombination, such as molecular cloning, to bring together genetic material from multiple sources, creating sequences that would not otherwise be found in biological organisms. This definition can be boiled down to taking one piece of DNA and combining it with another strand of DNA. First, we need some vocabulary. A plasmid is a circular self-replicating form of DNA found in bacteria. It's important to notice that this isn't the chromosomal bacterial DNA pictured here in red. There are also enzymes. You might have encountered this term with other biological processes, but here, enzymes act as catalysts to specific DNA sequences. This process of DNA cutting is also called digestion. To help understand how enzymes work, you could think molecular specialized DNA codes. Before we get into molecular level studying recombinant DNA, check out how recombinant DNA is made with this virtual lab bench activity. Pause the video, connect to this website, select bacterial transformation from the main menu. You've seen the lab bench steps to create recombinant DNA, but let's zoom into their molecular level. This is an overview of the process, but we'll go through step by step. First, the target gene from the human DNA, in this case human growth hormone, is isolated. A bacterial plasmid is extracted from a bacterial cell. Enzymes are applied next. The enzyme here is EGO-R1. Remember, enzymes act as scissors, cutting the DNA at a specific sequence. The cuts from the enzyme create sticky ends, or overhangs in the DNA. The sticky ends on the human gene fit into the sticky ends on the plasmid because they were cut by the same type of enzyme. The sticky ends of both
My friends, please attention. I will send you all clips in your WhatsApp group. Please only attention to this and let me know if you have any question. دوستان تمام این فیلم ها رو برای شما خواهم فرستاد. فقط لطفاً به مواردی که توی این فیلم ها داره گفته میشه نگران فیلم ها نباشید. من برای شما
This process is called DNA recombination, and the plasmid product is recombinant DNA. The recombinant plasmid is inserted into a bacterial cell. The bacterial cell will clone itself many times over, therefore making many copies of the recombinant DNA plasmid. Here's an overview again of the steps we covered. Pause the video and go over the steps again to make sure you understand the process. Recombinant DNA has been important in the medical field. An example of this is insulin. Before 1977, the only source of insulin was through animals that we found to be compatible with humans. This was an expensive and risky undertaking since people could have allergic reactions to animal-based insulin. In 1977, Dr. Riggs, Dr. Itakura, and Dr. Boyer created human insulin that was reproduced by E. coli plasmids. Today, the vast majority of insulin is produced by using recombinant DNA. Let's review what we learned. Recombinant DNA is DNA that has been combined with another piece of DNA. The process to create DNA first involves using an enzyme to cut the plasmid and target gene and create sticky ends. The sticky ends of both parts combine, forming a plasmid that has recombinant DNA. The plasmid is inserted into bacterial cells where the cell can replicate in laboratory conditions that you saw on the virtual lab bench. Recombinant DNA can help create synthetic drugs, including insulin. For an extension activity, investigate other medical and scientific research benefits of our DNA and write a small report on your findings. Today, we'll talk about the process of creating recombinant DNA. This lesson was made by Tyler Hogue and Mariah Thrush as part of Ohio University's NSF-funded Boat of Knowledge in the Science Classroom. What is recombinant DNA? Technical definition is DNA molecules formed by laboratory methods of genetic recombination, such as molecular cloning, to bring together genetic material from multiple sources, creating sequences that would not otherwise be found in biological organisms. This definition can be boiled down to taking one piece of DNA and combining it with another strand of DNA. First, we need some vocabulary. A plasmid is a circular self-forming DNA found in bacteria. It's important to notice that this isn't the chromosomal bacterial DNA pictured here in red. There are also enzymes. You might have encountered this term with other biological processes, but here, enzymes act as a catalyst to cut specific DNA sequences. This process of DNA cutting is also called digestion. To help understand how enzymes work, you can think of them as molecular-sized scissors sized to specific DNA codes. Before we get into molecular levels of creating recombinant DNA, check out how recombinant DNA is made with this virtual lab bench activity. Pause the video, connect to the bacterial transformation from the main menu. You've seen the lab bench to create recombinant DNA, but let's zoom into their molecular level. This is an overview of the process, but we'll go through step by step. First, the target gene from the human DNA, in this case human growth hormone, is isolated. A bacterial plasmid is extracted from a bacterial cell. Enzymes are applied next. The enzyme here is EcoR1. Remember, enzymes act as scissors, cutting the DNA at a specific sequence. The cuts from the enzyme create or overhangs in the DNA. The sticky ends on the human gene fit into the sticky ends on the plasmid because by the same type of enzyme. The sticky ends of both the plasmid and the gene come together, forming a complete plasmid. This process is called DNA recombination, and the plasmid product is recombinant DNA. The recombinant DNA plasmid is inserted into a bacterial cell. The bacterial cell will clone itself many times over, therefore making many copies of the recombinant DNA plasmid. So here's an overview again of the steps we covered. Pause the video and go over the steps again to make sure you understand the process. Recombinant DNA has been important in the medical field. An example of this is insulin. Before 1977, the only source of insulin was through animals that we found to be compatible with humans. This was an expensive and risky undertaking since people could have allergic reactions to animal-based insulin. In 1977, Dr. Riggs, Dr. Isakura, and Dr. Boyer produced human insulin that was reproduced by E. coli plasmids. Today, the vast majority of insulin is produced by using recombinant DNA. Let's review what we learned. Recombinant DNA is DNA that has been combined with another piece of DNA. The process to create DNA first involved using an enzyme to cut the plasmid and target gene, and then create sticky ends. The sticky ends of both parts combine, forming a plasmid that has recombinant DNA. The plasmid is inserted into bacterial cells, where the cells can replicate in laboratory conditions that you saw on the virtual lab bench. Recombinant DNA can help create synthetic drugs, including insulin. For an extension activity, investigate other medical and scientific research benefits of our DNA and write a small report on your findings. Today.
Okay, my friend. Do you have a question about recombinant DNA? Yes? If you have, please let me know.
These cats can glow in the dark. Now, is this a naturally occurring trait due to a random mutation? Nope. This is an example of genetic engineering, and we're going to take a look at how this process works in this video. Note that genetic engineering goes by different names. You might also hear it called gene splicing, recombinant DNA, or genetic modification. But all of these terms mean the same thing, which is basically this: taking DNA from one organism, combining it with another organism, and getting recombinant DNA, DNA from two different sources. Now I glow in the dark. Most it came from the cat, but the CG jellyfish that allowed it to work. Now you might be wondering how to combine DNA from a cat with DB jellyfish. Those are so many different. Well, the key is that the code is universal. So to take a look at the DNA of this bacteria and the DNA of this little biscuit, some differences, but they're all made of T, C, and G. Now the sequence might be different. We're speaking the same language, and because the genetic code is universal, engineering is a reality. And then there's another enzyme known as ligase, and its job is to glue DNA back together. Now the restriction enzymes in a little bit more detail. There are many different restriction enzymes, and each enzyme is specific for a particular DNA sequence. So this restriction enzyme will only unite when it sees the species TGG. This enzyme will only cut when it sees the sequence TCGAA. And this enzyme will only cut we've got our sticky ends. Now let's take a look at how these two enzymes together to create recombinant DNA. So here's our DNA sequence. Here's the restriction site for a particular restriction enzyme. First thing we're going to do is add a restriction and cut it into fragments. There we go. And here you can see the sticky ends. Now, we're going to add DNA from another source that's also been cut using the same enzyme. Now, this is important because using the same enzyme means that you get the same sticky ends. So here's DNA from one organism, here's DNA from a different one, but same sticky ends. Now, these fragments are going to stick together because of complementary base pairing. So here we go. This sticky end was complementary to this sticky end, and this sticky end was complementary to this sticky end, so they naturally came together. However, we need a little ligase at this point because ligase will help attach the sugar phosphate backbones, which are more difficult bonds to make. So once ligase gets involved, here we go. We now have our recombinant DNA. DNA from one organism combined with DNA from another organism. So this is how we would put two different genes together. But at this point, it's pretty useless. All we have is a recombinant DNA molecule. In order for that to actually build a protein that makes a trait, we need to put this DNA into an organism. So now let's take a look at the whole genetic engineering protocol. And we're going to use a particular example, a very commonly used example of genetic engineering. And this example involves the use of bacteria to be human insulin factories. That's right. Because of genetic engineering, we can make bacterial cells produce insulin that's not bacterial insulin, but it's human insulin. We can bottle that insulin and give it to diabetics. So here's how it works. First of all, let's start with the bacterium. Why would we use a bacterium as a human insulin factory? Well, one reason has to do with its genetic structure. Bacteria have a big circular cell, but then they also have smaller loops of DNA called plasmids. And it's very easy to move plasmids into one advantage to using bacteria. The other advantage is that bacteria are unicellular and they do asexually, which means that in a very short amount of time, you can get millions and millions of identical bacterial cells, clones. And this is important if you want factories. So let's see how the experiment works. We're going to start with our E. coli bacterium. Here you can see its chromosome. Here you can see its plasmid. And then we've got a human cell. And this human cell contains a nucleus, which contains DNA. And within that DNA is a gene for producing insulin. Now this is the correct gene. This produces good insulin. So the first thing we're going to do is we're going to take the plasmid bacteria, and we're going to take the DNA out of the human cell. Easy enough. Then we're going to cut both of them with the same restriction enzyme. So here the plasmid has been cut, and here the DNA has been cut. And we're going to use the restriction enzyme that cuts at sites around the human insulin gene. So it's important to pick the right restriction enzyme for your experiment. So now we have our human insulin gene, and we've got our cut plasmid. Now we're going to combine those two together. And because of complementary base pairing and because of the sticky ends, they're going to naturally bond together. Then we're going to throw in a little bit of ligase to help seal the sugar phosphate backbones. And now we have our recombinant DNA. Most of the plasmid is bacterial DNA, but it now has the human insulin gene. Pretty cool, but again, this is useless unless we put it inside a cell so that it can be used to make the protein insulin. So let's do that next. Step number 7. We're going to stick this recombinant plasmid back into the bacterial cell. And this process is known as transformation. Whenever you give a cell new DNA, it's transformation. So now we're going to let this bacteria cell reproduce. And in a matter of days, we'll have millions and millions and millions of identical bacteria, all of them with this recombinant plasmid that contains the gene for human insulin. So we're going to let these bacteria produce human insulin. And then what we're going to do
Okay.
Now, is this a naturally occurring trait due to a random mutation?
Okay, my friend. I think you have enough information about gene cloning, okay, and the basic of the gene cloning and the principle of the gene cloning and recombinant DNA technology. But you everything know you should know about the bacteria, and bacteria are microorganisms that are very important for biotechnology techniques. But what are bacteria? And in this section, I'm going to show you everything about bacteria, and I'm going to explain bacteria.
First step in cloning a gene is to isolate the DNA from the organism that contains the desired gene. The isolated DNA is purified and then fragmented with a restriction enzyme. Restriction enzymes used in cloning produce staggered cuts in specific sequences in the DNA, generating fragments with cohesive ends. Each fragment has a single-stranded sequence of nucleotides on its ends that is capable of hybridizing with DNA that has been fragmented with the same restriction enzyme. The DNA fragments are then incorporated into plasmids. The type of plasmid used for cloning has a single restriction site, and when cleaved by the restriction enzyme, generates the same cohesive ends that are in the fragments of the DNA to be cloned. The cohesive ends of the plasmid and DNA fragments now line up, and enzyme DNA ligase is used to form phosphodiester bonds. The next step is to incorporate the plasmids into bacterial host cells by transformation. Each cell contains a different segment of DNA from the original organism. Taken together, these cells represent a DNA library. The cells can now be placed in agar medium. The colony of cells containing the desired cloned gene can then be identified and isolated.
Okay, as I say in this last season, and based on different clips in this season, you know that the bacteria are very important in biotechnology, and bacteria are very important microorganisms in biotechnology, and help biotechnologies to cloning different genes. In the next season, I teach you everything about bacteria, their structure, and their genetics. If you have any question about restriction nucleases, DNA ligase, and gene cloning, let me know, and I will answer to your questions. Do you have a question about gene cloning, DNA restriction enzyme, DNA ligase. You should know everything about these information. بچه الان میخوام در مورد باکتری ها صحبت کنم. Okay? No, I want teaching everything about bacteria. Please attention.
Prokaryotes are the smallest known cells and the simplest in structure. The bacterial chromosome is a single circular molecule. It occupies a region of the cytoplasm called the nucleoid. The tiny volume of cytoplasm is continuous with the region of DNA. Ribosomes give the cytoplasm a grainy appearance. They are the molecular machine which polypeptide chains are assembled. The plasma membrane mediates the flow of substances to and from the cytoplasm. It has protein channels, transporters, and receptors for signals and substances. Most bacteria have a semi-rigid or rigid cell wall that surrounds the plasma membrane, supports the cell, and imparts shape to it. A protective capsule of jelly-like polysaccharide is the outermost layer of many bacteria. Villi are protein filaments that help cells attach to various surfaces and to one another. Flagella allows some prokaryotic cells to move about in a fluid environment. You may now review prokaryotic structure by clicking on the labels. A thick, protective capsule of jelly-like polysaccharide is the outermost layer of many bacteria. Most bacteria have a semi-rigid or rigid cell wall that surrounds the plasma membrane, supports the cell, and imparts shape to it. The tiny volume of the cytoplasm is continuous with the region of DNA. Ribosomes give the cytoplasm a grainy appearance. They are the molecular machinery upon which polypeptide chains are assembled. Villi are protein filaments that help cells attach surfaces and to one another. The plasma membrane mediates the flow of substances to and from the cytoplasm. It has protein channels, transporters, and receptors for signals and substances. The bacterial chromosome is a single circular molecule of DNA. It occupies a region of the cytoplasm called the nucleoid.
Okay. My friends, if you have a question about bacteria and gene cloning, please let me know. If you have not question, I will continue this lesson in the next season. Please let me know if you have any questions.